Perfect. Thank you. So first of all, yeah, thank you for the introduction, and thanks again for the nice invitation to speak today at this webinar. As I was briefly introduced before, so I'm Ecco Vacca from the Joint Research Center of the European Commission. And as expertise and background, I'm more a carbon cycle scientist, and I worked quite a few years on measuring carbon cycle and carbon fluxes, actually, mostly with eddy covariance techniques, and then using proximal and remote sensing for scaling up these fluxes, and with the main idea of trying to understand the interrelation between carbon fluxes and climate, in particular, different time scale. So today, I will set the scene a bit for the talk of Tommaso later, and I will give you an overview on why we use proximal sensing as a tool for better prediction of gross ecosystem productivity. I apologize in advance if I'm going to be a little bit simplistic on certain part. I wasn't completely sure about the overall expertise of the audience, so I tried to be relatively broad and relatively, let's say, simple in terms of concepts. And then, of course, Tommaso will actually dig into more technical aspects later on. So the outline of the presentation. First of all, in particular for the people overseas, I thought to give you a bit of an idea of what the Joint Research Center of the European Commission is, for the one of you that don't know it. And then I will go into the first part of the talk, which is about why we need remote sensing and proximal sensing to improve carbon cycle monitoring and modeling, and particularly the gross ecosystem productivity. And I will give two perspectives from the global carbon cycle perspective to the new perspective, or relatively new perspective of monitoring, reporting, and verification of nature-based climate solutions. Then I will go a bit more into the details of the interrelation between optical remote sensing and gross ecosystem productivity, and what are the gaps and uncertainties. So, but first of all, let me briefly introduce the Joint Research Center. So we are a Director General of the European Commission, under actually the Commissioner for Innovation, Research, Culture and Education and Youth. And there are 6 institutes in Europe. I'm located actually here in Ispra, in the, let's say, in northern Italy, right on the border of with Switzerland. If you're aware about the region between the Maggiore Lake and the Como Lake, so it's more or less around that area. But we are 6 institutes in Europe, and what we do is research to support policies. So we don't have a policy agenda. So actually, we're not a policy DG, but we do scientific research to actually provide data and knowledge to the policymaker to do the different, to implement the different policies. So in principle, we produce databases, research programs, and we have large-scale also research facilities to do this this task. And if you're interested in having more details, feel free also to drop an email, and I'm happy to answer. So let's go now to the topic of the presentation. I start a little bit far from the actual problem, but on the link between proximal sensing, so smaller scale, and gross ecosystem productivity. But I want to start from the perspective of the global carbon cycle, and why we need to improve our capability to monitor and model the global carbon cycle. I think many of you know this plot on the left side is the plot from Frigansin et al. This is the 2023 paper, but there are also several papers in the last years that also show somehow this kind of plot. And here on the left side, you see the net CO2 fluxes from the land. Positive sign here means a sink, because we are talking about land sink. But what is interesting here to see is actually the spread that you see between the different lines here, and the spread is actually the different lines are the different dynamic global vegetation models that are actually used to produce this sort of estimates. So models are different because they have different parameterization of vegetation in time and space, and they also have different way of describing processes. There was a very interesting review a few years ago in New Phytologist. I really suggest you to go to look at this paper if you're interested in the topic, that actually identified a series of steps that we can undertake as a community to improve models. And one specific aspect of this review was actually touching the part that on the relative transfer of the light into the canopy. That is, of course, a fundamental driver of gross ecosystem productivity. And it's something that in this kind of models is relatively, let's say, simplistic, or was at the time relatively simplistically described on the one hand. And on the other hand, with the fact that there is an increase of satellite products and proximal sensing monitoring tools, it's actually fundamental to develop models that are adapted to ingest this information, to better parameterize the model, or to directly use this information to drive the models and to improve the gross ecosystem productivity estimates. One aspect that is also important for today is that that I want to also underline is that, and it's a bit more with my belief, but I think that remote sensing is a potential and an amazing tool to really constrain gross ecosystem productivity. If we want to go to net ecosystem productivity, so also including the respiration terms, there is still a lot of work to do. And probably this should pass also through a bit of modeling and not only remote sensing. But in general, already constraining the gross ecosystem productivity would be great to improve the carbon cycle description. So, beside this global perspective also, as you know, there is an increased interest into nature-based climate solution. Here in Europe also we call it somehow carbon farming. And the point here is that through better management of land, reforestation, better agricultural management, the idea is that we can create ecosystems that tend to sequester more carbon, so it could be a solution in terms of carbon sequestration, but also more resilient to climate variability, in particular to extremes. And in this perspective, the combination between proximal sensing and flux tower is actually the key, because it's actually the scale where we can relatively well measure carbon fluxes and also optical properties, and learn how to transfer this information or how to use remote sensing to have then at a larger scale credible numbers in terms of the effect of this nature-based climate solution. This is also a lot of the work we have been doing here at the JRC to develop these kind of tools. Now, let's move a bit on why remote sensing is good to constrain gross ecosystem productivity. A couple of years ago, Zheng et al. Published this very nice review that I would suggest you to look if you're interested in the topic. Where, and one plot is this classical plot on the reflectance of vegetation, so the green line here, and the electromagnetic spectrum. And so here, in this plot, you really see very well why optical remote sensing it's used to monitor gross ecosystem productivity, is because it gives a lot of information about pigments, about cell structure, and also somehow leaf structure, and also the water content in the leaves, which are altogether important drivers of photosynthesis and therefore gross ecosystem productivity. So in the reflectance signal is really encoded all what we need to monitor photosynthesis and gross ecosystem productivity. And Tommaso later will also speak about fluorescence, which is also an important signal. And of course, many of the older but also the newer satellite missions are also targeting specifically these bands to have information about vegetation. So if we move from the, let's say, spectral domain to the temporal and spatial domain, here is where we are. So with proximal sensing, we can have information in terms of spatial scale, let's say from millimeters to some 100 meters. And at the temporal scale of few minutes with handheld probes to months and years with tools or with spectrometers like the ones that JB Spectra is developing on that are able to stay in the field and to measure continuously the spectral properties. And this is actually the interesting part, because this is the scale exactly at which eddy covariance can measure, and the scale where we can connect the two measurements to have a good description at very different time scale from let's say half hours to hours to weekly to even decadal now in some sites, sensitivities of fluxes to climate, and then connect that with the spectral information. Now, this is very nice on paper, and there are also quite few studies already showing the potential of doing that. But of course, there are some challenges, and I know Tommaso will dig into that. But these are also nicely described in this communication from Zoe Perrat. And the challenges are that, in principle, we are still building networks to connect these two measurements. And while the eddy covariance also did a lot of work on standardization and of data acquisition, but also post-processing, there is still a lot to do, but we're already in a good shape to have data sets and to do synthesis. When we speak about the use of proximal sensing and remote and eddy covariance together and do synthesis works, here we are still in the phase where we need to better connect the different measurements, understand the difference between different measurements, understand for example, the differences between the proximal sensing footprint, so the area that we see with the spectrometers compared to the area that we see with the eddy covariance, but also the differences that we have in results when we put together different sites where the measurements protocols were different. And this is something that is key, and that probably is something that we need to discuss also today, how to move forward in this respect. And it's a bit of the challenge to then try to do synthesis and also to use this data also for upscaling. Then I want to give you a bit my perspective on how we can use optical remote sensing for improving gross ecosystem productivity. And I try to summarize this into three, let's say, bubbles here, and I put few papers, but I want to say that I was not at all comprehensive. There is much more, but I just want to not overflow you with citations, just give you some key citations in my opinion. So one way is to directly use optical data, vegetation indexes, but also now with machine and deep learning, also we can use the whole spectrum, also together with climate data, to directly predict gross ecosystem productivity. This is something that, recently with also developments in AI or in general machine and deep learning, it's something quite often done. There is a lot of potential on that. There is also a lot of potential on using explainable AI to better understand the sensitivities of the bands to the fluxes and vice versa, so how they are interrelated. Of course, the question is how much still this can be completely generalized, but it's something that the community is working on. Then another aspect where actually in particular my former team in Jena, the MPI, was working a lot on using optical remote sensing to constrain relevant parameters for the vegetation models, like plant traits parameters, chlorophyll, VC max, so carboxylation rates, or also monitoring vegetation attributes, management. Very important to understand the actual fluxes, disturbances, and use this information then to better constrain the models and to have better estimates of gross and net productivity of the ecosystems. And then another aspect here that is quite promising, in particular if we develop models or a terrestrial model or land surface models that have a good radiative transfer routine, is to directly ingest remote sensing and proximal sensing information into these models in a more comprehensive data simulation scheme, Again, in this way we can improve the parameters, but we also can go a bit in the direction of what the climatology community does, also for climate forecast. So really adapt model to ingest the radiance and the electromagnetic signal, and to use that to improve the prediction of fluxes. So this is a bit the three, let's say, pillars of research that I see as very relevant for also for the talk of today. Of course, they are quite interconnected, but I think there are the three lines where we can actually move to progresses. Uncertainty and gaps. Again, I refer to Zeng et al. 2022, but if we talk about proximal sensing, I would say that mostly is this part below that is interesting. There are three aspects that are quite important. One is the scale effect. Again, I mentioned before the difference between the footprint of the eddy covariance, so the source area that we see with eddy covariance, and the proximal sensing footprint. This is very important. And also, when we want to scale up this to an ecosystem or even to a landscape, here there is a lot of uncertainty on the fact that the relationship that we see in at a specific spatial scale, actually between, for instance, GP and reflectance is actually maintained. So this is something quite important. There is complexity. There are different type of vegetation. There is soil that you can see here. Very important because affect quite a lot the signal. And there are these directional effects. So basically the geometry between the sun, the target, and the sensor can change. And this is a massive effect on the reflectance that typically we want to remove to understand better the physiological signal that is encoded in the spectrum, and then to really give information to better understand the photosynthesis. So those are three aspects that you need to consider quite carefully also in the different analyses. Then there is another aspect. So before I mentioned, we want to get rid of this directional effect that typically depends on vegetation structure to separate the physiological effect. This is very important. On the other hand, vegetation structure itself has a fundamental importance in determining for instance, the productivity rates, the potential productivity rates, but also it encodes somehow the signal of the forest management, if you talk about forest, of the age of the stand, and also the certain hydraulic properties that are important to understand also the efficiency of using water. So we saw in few studies that actually vegetation structure is key to predict, in particular, height and biomass, and of course, leaf area. Key to predict also the photosynthesis. So we cannot completely neglect this aspect, and we cannot completely just look at physiological signals that are related to photosynthesis, but we need to embrace the fact that vegetation as a structure and structures determine physiology and function. To do that, I think there are now valuable tools from the ground again. So the scale where we can link with remote sensing, we can have with proximal sensing, we can have a terrestrial LiDAR. For example, there are also some radar radiometers at the scale of the eddy covariance that can be used, and also if we scale a bit at satellite level, we have spaceborne LiDAR and also radar data that together can be used together with the optical signal to better constrain photosynthesis, because can carry, as I said before, information about hydraulic limitation, management, canopy architecture, structural diversity, all these aspects that are key to interpret photosynthesis and to predict actually photosynthesis. So, and with this I conclude, and I leave then the space to Tommaso with the following bullet points or take-home messages. So, proximal and remote sensing is actually one key tool, and I believe that all of you know, and is let's say supporting of this statement to constrain the carbon cycle, and also for monitoring, reporting, and verification of the nature-based climate solution, which is actually key and important, also for the private sector. As said before, there are key remote sensing and provide the key information to improve process model parameterization in space and time, in particular vegetation traits. directly estimate, to provide direct estimates of productivity, in particular with deeper machine learning, but also to use model for better data assimilation with a more dynamic description of the vegetation that can be also quite useful for certain applications. Of course, when we want to combine proximal sensing and remote sensing, proximal sensing, remote sensing, and eddy covariance, we need to keep in mind a few specific aspects. One is the fact that we really need to build a community and a network with consistent protocols and data measurements for synthesis studies. This is fundamental. And probably we should really try to not neglect vegetation structure to better describe photosynthesis. And with this, I leave the space for and the floor to Tommaso. And here are my contacts and my ex-account in case you want to contact me. And thanks again for inviting me.
Okay, so I am Tommaso Iulita from JBA Spectral Devices, and first of all, I want really to thank the LI-COR team for the invitation to this webinar, and Mirko for the presentation before. So this talk want to be a sort of follow-up of the talk of Mirko, focusing more on the real measurement that we can make at field scale with proximal sensing. So I started the presentation with this pretty cool picture in my view, which is depicting a site in Australia, Tambaramba site, and probably is giving the idea of where we are when we are measuring ground data with proximal sensing. So in this case, we have an instrument installed at the top of the tower, which is looking down together with the other sensor, which is equipped the tower with, for instance, eddy covariance sensor. And the idea is that we are measuring optical properties with sensor that are similar in terms of characteristics with the one at satellite level. So what we are doing is try to acquire high frequency temporal domain data to find a link between the optical properties we can measure with, in this case, spectrometers and the fluxes, for instance. And what we're measuring in, as already Mirko was showing, with field spectroscopy is basically the reflectance, and the reflectance here you see the spectral reflectance. So you have in the x-axis the wavelength, and you see different color curves which are representing different condition of the vegetation. So the reflectance is basically the ratio between the reflected light from the canopy, from the vegetation or the target that the instrument is measuring, and the incoming radiance that is reaching the top of the canopy, so the solar irradiance. And with this ratio, we can basically get out most of the influence of the light change condition, and we can really try to understand this property, the optical properties, which are often linked with biochemical or biophysical aspect of the vegetation. And in particular, as was already pointed out, there are different regions of the spectrum which are affected by different aspects of the vegetation. For example, here we are in the visible domain, so basically in between 400 and 700 nanometers, so the red, green, and blue region. And this is affected by the chlorophyll concentration. And in fact, you can see, for example, in the green canopy, you can see this absorption, which is not evident any longer in the senescent canopy because this chlorophyll concentration changed. But it's not only that, there are other regions of the spectrum which are sensitive to some other physiological processes, for example, the xanthophyll cycle. In this case, the region is tinier and more into the in the green region. Then there are the near infrared, which is more related to the structure. We have information about leaf water content. And last but not least, we have also the fluorescence signal, which is visible in the reflectance. Actually, fluorescence is not really reflected light, but is light which is emitted by the chlorophyll during the photosynthesis. And you can see it is very tiny signal, which you can appreciate here in red in the reflectance shape. And this is something which is again related to the physiology of the vegetation. And traditionally, this information, you can see it contains a lot of important aspects related to biochemical, biophysical, physiology, phenology, information of the vegetation. And there are ways to synthesize this information, for example, vegetation indices, so spectral indices, which are computed considering different bands positioned at different portion of the spectrum. Probably the most famous one is the NDVI, which is computed using the information on the near and the red region. So basically, it gives you an information about both chlorophyll concentration and structure of the canopy. But there are others which are more related to physiology, for example, the photochemical reflectance index, which is computed in this range, so where the xanthophyll cycle has an effect. So these are ways to basically synthesize all this information, which otherwise is a lot of information, to simple indices which are related to these different properties. And so what we're doing as a company is basically manufacturing field spectrometers, hyperspectral field spectrometer, which are then capable to work automatically in the field and to collect continuous data of radiance and irradiance. And this is basically most of the case used in a flux tower, because one of the main aspects and the main, let's say, scientific question that the user are interested in is really to find the link with fluxes. The company was founded in 2016 and is based in Düsseldorf in Germany. And these at the bottom are the three main instruments that we are manufacturing. So on the left, you have the flux, the fluorescence box, which is focusing on the passive measurement of chlorophyll fluorescence. Then we have the reflectance box, which is getting the reflectance in the visible near infrared region up to 1000 nanometer approximately. And then the most recent instrument is the NOx, which is basically an extension of the rocks covering up to 1700 nanometer. Quick focus on the flux. So as you see from the picture, the flux is basically a box with some holes that you can see here. And through this hole, we are pushing out some optical fibers, and these optical fibers are basically used to bring the light to the spectrometer. In the case of the flux, we have two spectrometers included. One is focusing on the fluorescence range, so from 650 to 800 nanometers, with a pretty high spectral resolution and a good signal to noise ratio. This is needed because fluorescence is a very tiny signal, so measuring it is pretty demanding in terms of performance of the spectrometers. Whilst the second spectrometer is a broader spectrometer with less restriction in terms of spectral resolution. In general, what we have is basically fiber looking up equipped with the cosine receptor, which are measuring the solar irradiance with a field of view of 180 degrees. So it's an hemispherical measurement, whilst the downward looking fiber has a fixed field of view of about 25 degrees. So it's like a cone, which is giving you at the end the footprint, so the area that you are measuring at. And the flux is basically continuously acquiring alternating measurement of incoming radiance and reflected radiance. And so you can see the bottom left an example of the measurement that you can get. This is cut by the spectral resolution of the fluorescence spectrometer. So is just focusing on the red edge region, let's say. And basically what you can get out in this case is a time series of fluorescence of 7 years of installation of the flux in the same place, the same location. This is a typical, in this slide, I just want to show you some typical example of installation. In some case, tall towers, in some other case, tripod at the ground. This really depends on the target. In some other case, some more creative ways of installation, carrying the instrument on the shoulder. And on the sketch on the right, you can see basically what we are measuring. So if we are installing the instrument at the top of a tower over a forest, we are increasing the distance between the sensor and the canopy, so the footprint of the instrument will be larger. And consider that approximately at 50 meters, we have a footprint of 20 meters. So it depends then when what you want to measure. And also this information is a key information when you are setting up your measurement, because then if your field has some degree of heterogeneity, you need to properly set it up, let's say. And in general, with this slide, just wanted to show you that we're not coming out of the blue, but this instrument is coming out from a pretty long research period, in particular under the framework of the European Space Agency, and in particular the Fluorescence Explorer mission, which is a mission that is expected to be launched in 2026. And during the preparation phase of this mission, at the beginning, there was really a lot of field campaign to test the concept of the mission, how we can measure this signal, and starting from manual instrument up to prototypes that slowly were making automatic measurement and so on. And at the end of 2016, we thought that we had an instrument which was mature enough to start the commercialization, and we started actually with the flux instrument. And since the beginning, the last year, we in this map, I just want to give you a sort of overview of the location of the instrument, how they are spread all over the world. We have about 65 flux system, about 40 rock system, and some of these new NOx system also now operational in the field. Consider that most of these are used coupled with eddy covariance tower, because again, one of the main interesting point is really try to collect high temporal resolution measurement and link them with the GPP. In terms of standardization, what we are providing to the user is basically a data processing pipeline, which is entirely written in R, so is open source code, and most of the functions are already published in GitHub. And the idea is also to facilitate the user, and we develop a graphical user interface where you can just select few input and use it for processing year of data. And also making some data filtering, data aggregation at different temporal scale of aggregation in order to eventually match the data time scale of eddy covariance, which is typically alpha hourly. The concept is of the data processing is basically starting from the raw data, which the instrument is collecting. Together with the calibration we are providing, the calibration coefficient, you can convert data to radians, and the ratio of the two is basically giving you the reflectance. In this case, you see a diurnal course of reflectance with the V's near, so the broad spectrometer. And then from that, you can basically compute all the vegetation indices you are interested in, or use the reflectance directly, and retrieve fluorescence. And together with this, in the processing, we are also providing some quality flags, which can be used for filtering out the time series. For example, if the measurement is collected under unstable illumination condition, which is something which can affect the results. And after that, this data can be used for the temporal aggregation. So what we did was really starting to make something standardized. Also, as we heard by Mirko, the importance of standardization in order to make this data available also in networks. And what also we did was this collaboration that we started with LI-COR, and we made since 2023 all our devices compatible with Smart Flux. These to facilitate real-time visualization of the data. In particular, all the process or part of the processing is made inside the instrument, inside the flux, ROX or NOx, and some of the output, in particular up to 9 variables, can be streamed out to Smart Flux, which then can be used for real-time visualization using, for example, a Flux Suite. And this, I think, is something that is really facilitating also the user. And it also go into the concept of facilitating the processing, getting this standardized processing to enable the usage of this data into networks. And talking about networks, I think this is really a key challenge of upcoming years. And as Mirko said, Flux community has a very long history of network. In the field spectroscopy community, this is not so consolidated. There are some examples of existing network, and this is, for example, SpecNet, which is a US-based association. Here I reported also the link if you want to have a look at that. And the idea of this network is really try to merge different sources of proximal sensing data with the goal, again, of better understanding how to scale up this information for carbon prediction and water vapor fluxes in general. But what we are trying to do is something a bit, try to basically make our instrument and the output of our instrument compatible with the existing Flux network. Because at the end, I think that the final user that could really make use of the data we are collecting is in part the Flux community. And so what we are trying to do is really spending time to consolidate the metadata, the data processing, the data format, in order that this can really be something in the direction of joining existing Flux network. And we started the collaboration with, in particular with ICOS and FluxNet in order to do that. If you are familiar with these networks, you know how high standard they have in terms of metadata and data processing. So this is something we are working on. We have few sample data already available, in particular the European Fluxes database cluster, and it's something that we will continue working in the coming year for sure. Then I picked up this slide from Mirko, where it was showing how this optical remote sensing can really play a role for a better vegetation productivity estimate. And in particular, I'd like to focus more on the part where most of our instruments are used for these activities, and these rely mostly on the empirical relationship between GPP and vegetation indices. Again, so what we are measuring is the reflectance, and this has a lot of information encoded on it, which can be used for this link with GPP. One aspect that I will focus more on is really this fluorescence signal, so this emission of light, which is made by the chlorophyll during the photosynthetic processing. And because this is also something which really took off in the last year. And if you can see here, I put just a number of scientific publications about fluorescence and how this increased in the last year. And you can see that the increase was really massive. So this is giving some hope that we are going into the right direction, and that this signal is something that can be really exploited for a better understanding of the ongoing processes. And one case study I want to highlight is this one from Benjamin Deschamps, which was published in 2022. And I think it's a pretty cool paper because it's really following a upscale approach from bottom up. And the authors were basically investigating several sites equipped with a field sensor for measuring fluorescence and other variables. And what they did was really try to investigate how the fluorescence was correlating with GPP, proposing other indices as a better proxy in this case for GPP estimate. In this case is the near VP, which is a sort of NDVI normalized by the photosynthetic active radiation and multiplied by the reflectance in the near. And they were basically showing these different, investigating these trends at the different sites, covering different plants. So you see here, for example, the difference between C3 and C4 plants. And what they did also was investigating these at different scale, in particular from ground to, again, ground, but using imaging hyperspectral sensor from airborne, and then finally extending this information at global scale. And in this case, they were finding that the near VP was a promising, was found to be a promising proxy for GPP. Taking into account in particular some of the structural effect that Mirko also mentioned before in the fluorescence estimate, and that this is a promising proxy for next and improved GPP estimation at satellite level as well. Another case study I wanted to highlight is was published by Zoe Pirat, and in this case is also something pretty cool because it's using some simple light use efficiency model from Monteith in the 70s, where basically GPP is a combination of APAR and light use efficiency. And the good thing of this approach is that you can use optical remote sensing variables to estimate this single information. So it's pretty appealing from a remote sensing point of view. And in this case, they were using, they were investigating and analyzing 2 years of data over a boreal forest, try to understand which was the best predictor for GPP. In this case, they found that fluorescence was performing pretty well at different scale. This is the good thing of having also high frequent measurement at the ground, that you can then investigate the effect also of the temporal domain on this relationship. And they were finding, for example, that, in this case, that fluorescence was linearly correlate with GPP if analyzed at monthly or daily scale, whilst the relationship was changing, and the linear relationship was not preserving when looking at alpha hourly scale. And they were also making some consideration about what was driving this, so PAR and air temperature in particular. And this links to the next study. So this is a plot coming from a paper from 2014 on modeled data. In this case, the authors were trying to understand how the fluorescence efficiency and the photosynthetic efficiency was changing according to other environmental factors. And what they were showing is that fluorescence, if in many cases considered to linearly scale with GPP, this can also be not always true, because there are other environmental factors which can affect this, and in particular, temperature was one of the factors that was highlighted to affect this. And in 2021, David Martini was publishing this paper, where basically for the first time, also with real data measured in the field, this was in Spain, so a savanna-like ecosystem, and we could really notice that during an heat wave, this relationship was changing significantly, which was a good confirmation from what was shown in modeling studies. Of course, this posed some challenges in using this signal, because it's a complex signal, is something that is changing according to environmental stress occurring on the vegetation. So it's really pointing out the fact that in order to properly use that, we need to better understanding the process, and this is something that we can do with the field spectroscopy. This was, I think, the last case study I was putting. Again, there could be many more, but in favor of time, I was picking up only these ones. And then I want to show you one current study we are conducting at the moment. As Mirko was already presenting before, one of the problems when we are trying to link these two techniques, so field spectroscopy and eddy covariance, for sure we are then facing a problem, which is the footprint mismatch. As you know, so the field spectrometer are basically looking at one single spot, which depends in terms of size on the height of the sensor, whilst eddy covariance is providing you a measurement with a footprint which is changing during time according to wind direction, turbulences, and so on. And I think one very nice step that we are currently trying to make is to understand if we can make use of new technologies for analyzing eddy covariance data, which in particular is flux mapper, which here I reported a paper wrote by Stefan Metzger, which is now the CEO of Atmofax. And basically this tool provides you the capability of not only having the traditional, the classical eddy covariance footprint, which is changing across time, but it's basically used, squeeze all the information that is collected by the eddy covariance sensor at high frequency, in order to also give the possibility to exploit and to provide a flux map. And you can clearly see this in this pretty cool video, where the classical eddy covariance footprint is changing across time, but is just giving you one single value, whilst here it is really discriminating the fluxes from the different origin points. And there are different levels of output of this tool. Basic level is just using information from atmospheric information, whilst in the second level it is also using some geospatial information, and even further it can go to gap the fields that are coming from the second step, using even more advanced and enhanced techniques. And I think this is pretty cool, because we can really then try to match and to locate the measurement we are doing at the field scale with this flux map, and this is something we are currently testing, and hopefully we have some result to share soon. So this was the last slide, let's say, and here a few concluding remarks. As we have seen, field spectroscopy is a powerful tool and provides a link between in situ data and remote sensing. The spectra contains really a lot of information from chlorophyll content, structure, physiology, water, and so on. And it can really be used to improve our understanding of the processes at canopy scale, which is then possible to scale up to global. And in fact, is what we are currently doing, making this continuous measurement to really try to scale up the link that we find at the local scale. As was stressed out, there is really a need of standardizing instrument, data processing, but also metadata to make this data more friendly use to scientific community to join existing network, because this will also help in improving and to cover the larger variability of ecosystem types, so to better again understand the processes and how these can relate to different vegetation types. So what we, as Mirko was saying, also most of the studies are focusing on vegetation indices and SIF. What we can also do with this new AI potential is also exploit better all the hyperspectral reflectance information. And again, the structural effect can affect the canopy. There are ways to compensate to this. There were a lot of study on this direction, and this is something that is still ongoing also as to understand if this structural effect can be used as a proxy of something else. And finally, what I was just presenting briefly before, this new technology also from the eddy covariance techniques can probably be used to better match the in situ data with fluxes, which can also be used at satellite level, where in any case we will have a map of measurement. So yeah, I think that was all, and want to thank you for listening. If you want to contact me, there are also here the reference, and yeah, thanks again for the invitation.
Thank you for that presentation. We had some great questions in the Q&A while you were presenting, and I have just a couple that we'd like to pose to you here at the end. And then for anyone else who has questions, please go ahead and type those into the Q&A that you can find at the bottom of your screen, and we can get to those questions while we have time here. So first one I wanted to highlight for you, we had a question about setting the instrument at an angle to capture a larger view angle instead of vertically downward at possibly 45 degrees. So how would you answer that one?
Well, as also Mirko was saying, so we are measuring an optical signal, and the optical signal is affected by the structure. So if we are changing the angle you are measuring the signal, this can affect the measurement, because it is the so-called BRDF effect, so the bidirectional response function of the target. And typically the recommendation is to try to minimize the nadir view, so try to stick to nadir, because otherwise you can have effect. And there are publications which are really trying to measure with goniometer the effect of that on the signal, and you can see that there are some hot spots, some dark spot, and so the common practice is to try to stay closer to nadir. Then of course there are some logistic limitation in tower as always, so it can be tilted, but yeah, maybe 45 degrees is a bit too much.
Sure. Okay. Thank you. One other question we had was how different leaf layers affect the measured reflectance. Is that something you could address for us quickly as well?
I didn't get this. Sorry. How?
Sorry, how different leaf layers affect the measured reflectance?
Yes. Well, so actually what we are looking is the signal which is coming from this cone of light, and typically what we are looking at is the upper layer, and this is something that is also yeah, when I was mentioning the footprint mismatch, so actually the eddy covariance is giving you the information of the whole vertical profile, whilst what we are focusing on is mostly the upper part. And this is something that probably needs to be investigated more, how this is really changing. But it's also what you are basically measuring at satellite level. So the most influent part is the top of the canopy, and then really depends on the structure of the canopy, how dense it is, and how the light can really penetrate on it. But yeah, consider that most of the signal is coming from the top.
Okay, thanks. Here's another question that just came in. Are we able to get an indication or calculate the NPQ at the top of the canopy from a flux system?
This is basically ongoing studies. So there are publications which are currently using the photochemical reflectance index, which is mostly related to the scientific cycle, and this is triggered by, let's say, NPQ. So this is there are papers that are showing this NPQ can be tracked. Again, it really depends on the structure of the canopy, and the evolution, so the phenological evolution during the season, because this can also affect somehow the reflectance shape, and then this index can suffer a bit. There are ways to studies that are ongoing to how to minimize this effect to clean the signal in this direction.
Okay, great, thanks. So a couple other notes. We had some questions about using the system with our LI-COR instruments. And we shared a link to an application note in the Q&A for how to do that. I will go ahead and put that link also in the chat for everyone. So that's available if anybody wants to look at that application note. We also had questions about sharing the recording. So we will be sending the recording out to all webinar attendees and registrants following the webinars today. It might take a few days for the email to go out, but you should receive it sometime in the next week. Any other questions before we wrap things up? Okay, and if you do have questions, of course, you have our contact information, and Tommaso and Mirko were kind enough to share those in the presentations as well. Thanks to both of our presenters for the excellent information today and for being willing to join us for this webinar. If we have no other questions coming in, a lot of thank yous to both of you from the attendees, so fantastic. Let's go ahead and wrap things up and conclude the webinar. Thanks again, everyone, for attending, and to the two presenters for the information today.